Very excited today to welcome everybody to this will be our third edition third chapter of the Ratios Matter seminar series. And I'm really excited to introduce my really good friend, Nicole Wagner. So we met, we both did our PhDs with Paul at Trent University, and Nicole taught me everything I know about growing Daphnia and doing cool biochemistry stuff. She's done a few different postdocs now, making the rounds. So after Trent, she went to the U of T, did a lot of cool metabolomic work with Daphnia. Then she switched it up and went to Cornell, did some kind of weird quorum sensing, Nicole. I don't exactly remember. And then now she's down at Baylor, and she's doing all kinds of really cool work with Dad Scott. So she apparently just got tired of Daphnia, and now she's mostly focused on cyanobacteria. And she's also, I'm really proud that I think I might have been the first person to try to teach Nicole how to drive a boat, and she probably has more experience than I do now driving boats. So she's jack of all trades, really accomplished, has a ton of papers, and this I'm sure will have no problem getting a full-time gig in no time, and then that'll be in trouble. So all right, so I'm really excited to hear what Nicole has to talk about today. And yeah, Nicole, take it away.
All right, thanks Clay for that intro. And yeah, I can drive a boat a little better than when the first time. So yeah, today I'm going to be talking mainly about my postdoc research here on HABs and looking at stoichiometry and the ecophysiology and toxin concentration and cell quotas. And so, oh great, now it was working and now it's not. Sorry, y'all, technical difficulties.
I can see your mouse. There you go.
Yeah, there we go. My mouse was on my wrong. I have dual monitors, and it seems to be more complex than what it's worth sometimes. So yeah, I just want to first start off by acknowledging our NIH Oceans and Human Health Center that Baylor is a part of. We are part of the University of South Carolina Center, and the two awesome PIs that I am working with here are Brian Brooks and Thad Scott. And in the Brooks lab, they're sort of focused on more of the toxicology, bioaccumulation, and behavior and physiology of higher food webs, like particularly with fish. And then in the Scott lab, we're focused more on the stoichiometry and how that affects cyanobacteria growth, ecophysiology, and also the toxin cell quotas and concentration production. And that's what I'm going to focus my talk on today. And so HABs are common. So this is just the global distribution of microcystis. This is from a paper in 2016 by Harkey and others. And you will see that this map, there's a lot of green and blue on it, and there's very little white areas on the globe. So microcystis is not found in Antarctica, but all other continents of the globe with that exception, and very few white areas on this globe. And the white areas are places in which microcystis was not at least found in 2016 as possible. Since 2016, there is microcystis in some of these locations. And microcystis can produce a hepatotoxin called microcystin. And all the green areas on this map are where microcystin concentrations have been present with microcystis blooms. And the blue areas, microcystin hasn't been detected. And that could be because it just wasn't measured, or it actually wasn't a toxic cyanobacteria bloom. And so in addition to these HABs being really common, another common occurring theme is elemental imbalances are also very common in our freshwater ecosystems. So this figure is modified from Scott and others from 2019, published in L&O Letters. And they use the National Lakes Assessment database to look at sort of the stoichiometry of lakes, and they used it to look at sort of excess nitrogen. But just looking at more of the stoichiometry here. So on the x-axis, we have the total nitrogen in micrograms per liter on a log scale. And on the y-axis, we have the total phosphorus also on a log scale in micrograms per liter. And then using the Guilford and Hecky sort of limitation thresholds, where N limitation is less than N to P of 9 by mass or 20 by mole, and P, strictly P limited, is greater than an N to P of 23 by mass or 50 by mole. And those are depicted with the red line. For anything above the red line, it could be classified as N limited, and anything below the black line could be classified as could be P limited. So just looking at the 2012 NLA database, that comes to 24% of the lakes that were sampled were classified as phosphorus limited, 28% could be classified as nitrogen, and the majority of them are classified as co-limited, with 48% showing up between these black and red lines within the 9 to 23 N to P by mass ratio. And so the great thing about the NLA database, when they sampled, they also looked at cyanotoxins, particularly microcystin, and there's been numerous papers also published on that. And so this is a paper by Yuan and Pollard in 2017, published in Freshwater Biology. And so the X and the Y axis are the same here. And so we have total nitrogen on the X and total phosphorus on the Y. And the data here in the center are the microcystin concentrations, with low microcystin concentrations showing up in the white and really high microcystin concentrations showing up in the dark red. And so what they did is they generated a model to look at the likelihood of finding microcystin. And they found that this was really dependent on the total nitrogen concentration, more so than the total phosphorus concentration. And these contour lines are the probability of finding different concentrations of microcystin. So for example, if you look at the 10,000 micrograms per liter total nitrogen, or 10 milligrams per liter, it's around we're expected to find around 1 microgram of microcystin per liter in these areas. But it seems, and as you increase the total nitrogen, the likelihood of finding even higher microcystin concentrations increase. So, but does the stoichiometry, going back to sort of merge these two studies together, does the stoichiometry also influence these microcystin concentrations? So I pulled the 2012 NLA database, and I just plotted here the microcystin concentrations above 0.3 micrograms per liter. So in the blue squares, we have the microcystin concentrations between 0.3 and 1 microgram per liter. And then in the yellow circles, it's above 1 microgram per liter. And again, you can really see that a lot of these microcystins occur sort of in more of this co-limited range between an N to P of 9 and 23 by mass. However, when we just look at the average N to P of the lower microcystin concentrations, the average is 32 with a median of 26 by mole, and 14 or 12 by mass. But then when you get to greater than 1 microgram per liter, the N to P, average N to P is 37 and a median of 31. And so this really begs the question, if stoichiometry and cyanotoxins are correlated, because it seems like the higher the microcystin concentrations, we actually seem to be approaching more of these really high N to P systems. And if so, what are the relationships? And so sort of just to look at a common cyanotoxins that we have found in freshwater environments. These are 3 common ones. So we have microcystin LR, cylindrospermopsin, and anatoxin. And I highlighted the nitrogen molecules. So microcystin has 10, cylindrospermopsin has 5, and anatoxin has 1. And then if we look at the C to N of these, microcystin has a C to N of 5.7 or 14% nitrogen. Cylindrospermopsin is even more nitrogen rich. It has a C to N of 3.5 or 16.8% nitrogen. And anatoxin is actually relatively nitrogen poor with a C to N of 11.6 or 8% nitrogen. So then this sort of, we have these varying ranges of C to N of these toxins, but this really leads to the question of how plastic are cyanobacteria C to N ratios. And so cyanobacteria can be relatively plastic in their C to N composition. On the x-axis, we have different genera of cyanobacteria: Microcystis, Dolichospermum, Aphanosomena, and Cylindrospermopsis. And then on the y-axis, we have the stoichiometry with the number of samples used to make this graph also is at the N number. And so what's really striking here is besides Microcystis and Dolichospermum, there's not a whole lot of C to N information out in the literature. And a lot of the Microcystis and Dolichospermum I found were actually from our lab experiments. But what we found here is that Microcystis has a much wider range of C to N compared to the other 3 species that can actually use atmospheric nitrogen to fix, so it's harder for them to become nitrogen limited. And then on the horizontal lines are the different types of common cyanobacteria toxins. So just above the 10 is the anatoxin, and then the other ones, the blue, cyan, and orange, are microcystin, cylindrospermopsin, and saxitoxin. And the dotted line is the Redfield ratio, just to give everyone sort of a sense of common threshold ratios. And so what this really shows is, even though that normally cyanotoxins aren't a huge component of the cell, they actually are relatively high in N demand. And so therefore, especially in N-limited conditions, their production could actually cause these growth toxin production trade-offs. So this sort of led to the stoichiometric hypothesis of cyanotoxins. And this idea was formulated by a previous Ratios Matter editor, Dedmer, and colleagues in 2014, published in Ecology Letters. And they looked at a meta-analysis of PSP or saxitoxin that were produced in dinoflagellates, and also looked at MC, which is microcystin, and NOD is nodularian, that were produced in cyanobacteria. And they looked at how their cell quotas of these toxins responded to nitrogen and phosphorus limitation. So on the top, this is the response ratio to nitrogen limitation. And on the bottom here, this is the response ratio to phosphorus limitation. And they're comparing it against nutrient replete conditions. So if we see data that falls above the 0, you can say that the nutrient limitation actually increased the production of, or increased the cell quota of these toxins. And if data falls below the 0, it actually decreased. And so what they found was, under N-limiting conditions, these enriched cyanotoxins decreased. However, under P limitations, the dinoflagellates with the saxitoxin, their concentration increased, and overall they increased. However, the microcystin and nodularian look like they're decreasing. However, it wasn't significant. So then we wanted to sort of explore this further, which then brought us to our first set of experiments that we did, was what affects the growth stoichiometry and toxin production in microcystin. And this was published in 2019 in Toxins. And so what we hypothesize here is that microcystis will adjust their primary metabolism to meet their nutrient demands. So we have an elemental composition that consists of different amounts of nitrogen and phosphorus. The organisms will take up what they need for growth and reproduction. And then when they become limiting, they'll alter their life history. And this graph is just showing that the changes in growth or in vivo chlorophyll. And then the second hypothesis is that the nitrogen requirements for primary metabolism could potentially cause a trade-off between growth and secondary metabolic toxin production. So primary metabolites, or primary metabolism, this is controls growth, reproduction, and maintenance, whereas secondary metabolism, this is actually synthesized through non-ribosomal processes, and this is where the cyanotoxin production occurs. And so some previous studies, especially with plants, have looked at sort of this hypothesis that and found that enriched secondary metabolite production is sensitive to the N in the environment. And under N limiting conditions, you actually end up with decrease of N-rich secondary metabolites. So that's what we would hypothesize, or that's what we would predict. And so what we did is we grew Microcystis aeruginosa, Utex 2385, and we had a bunch of phosphorus levels. We had 5 phosphorus levels ranging from really low phosphorus, 20 to 700 micrograms of phosphorus per liter. And we generated 11 N to P ratios for each phosphorus level, ranging from 1 to 100. And so we added nitrogen to maintain the N to P ratio. And the nitrogen source we used was nitrate. And we grew the cultures for 21 days, and we saved for carbon, nitrogen, and phosphorus, and total microcystin that consisted of both particulate and dissolved fractions. And these are just 2 pictures of all the jars that this experiment contained, which there was many. And let's just jump right into the results. So here, this is the biomass, and on the first panel is the log nitrogen resource. This is how much nitrogen we put in the jar, and the second panel is the log phosphorus resource. And then the colors are the different phosphorus treatments. So we have the orange in 20, the green in 40, the blue squares in 80, and so on and so forth, up to the blackish triangles in our 700 micrograms of P per liter. And so what we found is that biomass increased until around 1900 micrograms per liter of nitrogen. Since this is a log scale, this is actually a segmented linear regression. And after the 1900 micrograms per liter, we found that actually the slight increasing nitrogen concentrations actually resulted in a slightly negative biomass relationship, being really driven by these really high nitrate concentrations. And then looking at phosphorus, we find this linear increase here. So the more phosphorus you add to the system, the more biomass you end up with. And all this variation around this line is caused by the different nitrogen concentrations. So then to look at sort of the C to N stoichiometry responses. So here we have the three panels. We have the response to just the nitrate environmental N resource that was found within the jar, and then looking at more of the the microcystin cell quotas and total concentrations. So just looking at this first graph here, so again, we have the log nitrogen resource on the x-axis, and then the C to N of the microcystis on the y-axis. And again, this is a linear regression, a segmented linear regression. And we find a really strong relationship here of decreasing C to N as our nitrogen concentrations range from around just under 10 micrograms per liter until just over 2000 micrograms per liter. And then adding more nitrogen ended up with more or less a stable C to N of just around 5. And then looking at the C to N and how that affected by the microcystin cell quota. And we can see there's this negative relationship that as you have more nitrogen, so a lower C to N ratio, you end up with higher cell quotas of microcystin. And then if we look at sort of the biomass, so what was in the jar, so not looking at the cell quota, you really see this strong linear decrease that the more nitrogen you have in the system, the lower C to N and the more total microcystin concentration you have. And then similarly, we can do the same thing with phosphorus. So here's the C to P on this first graph, and then by the log phosphorus resource ratio. So we were able to really change their C to P drastically. We were able to make them have a C to P ratio from just over 50 to almost 1,000 in our different phosphorus treatments. And we found this nice relationship. Obviously, as you add more phosphorus, you decrease their C to P ratio as expected. Within the cell quotas, between C to P and microcystin, we didn't find a relationship. It just sort of looked scattered. However, when you incorporate the biomass, you find a similar relationship to what we found with the biomass with the C to N, in that lower C to P ratios generated more biomass and it created more total microcystin LR concentrations. And then sort of just to look at the N to P by their environment, looking at the N to P in the microcystis and then the cell quota. And we're just going to quickly go through these one at a time, each of the phosphorus levels, just showing them one at a time to have a little bit easier understanding of what's happening. So this is the 20 micrograms per liter phosphorus. So we can see that these are positive linear relationships between the N to P in the bottle versus the N to P in the population of microcystis. And the cell quotas also have the same sort of linear positive relationship for the 20 micrograms per liter phosphorus. And then adding on the 40 micrograms per liter phosphorus, we find an even steeper positive incline between resource N to P and microcystis N to P, and a slightly more shallower N to P, but still positive regression between resource N to P and cell quota of the microcystin within the cells. And then adding on the 80 micrograms per liter, again, we see this nice strong relationship between the resource and the N to P and the microcystis. And again, a positive relationship between the resource and the N to P and the microcystin. And then when we get to 175 micrograms per liter phosphorus, we begin to see the N to P become more shallow. So it only ranged sort of between 20 and 40, but we did still see this nice strong increase in microcystin cell quota and versus NDP of the resource. And then as we continue to add more phosphorus, the NDP change is less, but we still see this nice strong relationship between microcystin cell quota and NDP resource. And then finally, similar results with the 700 micrograms per liter. And then, so I just want to take a couple minutes to explain what's going on. And so we're going to look at the NDP of the 75. And so this is microcystis phosphorus in this one graph that you can see. And on this this lower x-axis, this is the resource phosphorus. And so this 1-to-1 line is basically the phosphorus that we added. So if the data points fall on this 1-to-1 line, basically all the cells took up the phosphorus and there's no more phosphate left within the culture to be taken up. And if these dots fall below the line, then there still should be soluble reactive phosphorus for cells to be taken up. And basically we see that the 20 to 80, we actually do end up having phosphorus limitation, whereas concentrations higher than the 80, there's still a little bit of phosphorus when we took this experiment down. So there's not really a whole lot of phosphorus limitation happening, even though the NDP is 75, which would give an indication that it should be phosphorus limited. And then another point of evidence of this is just looking at the in vivo chlorophyll A data that we calculated through time. And so as there's more biomass produced and growth is continuing, we expect to see the in vivo chlorophyll A concentration increase, fluorescence increase. And again, we see here with the 20, it sort of decreased and leveled off, same with the 40 and the 80, indicating that there was limitation happening. However, in the 175, 350, and the 700, we still are seeing growth. And then just to look at sort of the microcystin cell quota, just at this NDP 75, there is some variation, but there's actually not a whole lot of significant differences. So even though we think that, even though it sort of was originally hypothesized that phosphorus limitation conditions can really promote these high microcystin cell quotas, we're seeing that even under phosphorus rich conditions can even produce the same amount of microcystin cell quota. And so just to wrap up this experiment, we found that basically biomass, when you add more phosphorus, you tend to get more biomass present, and toxicity was correlated with NDP, with high nutrient NDP ratios cause the cells to be more toxic. And this sort of was regardless of phosphorus limitation status. Now, this experiment, as I mentioned, was done in nitrate, and we know that nitrate's not the only form of nitrogen in the environment that's commonly found in lakes. So nitrogen can come in many forms. There's nitrate, nitrite, that's normally not very high, ammonia, or also urea. And these have different uptake pathways in cyanobacteria that require different amounts of energy. For example, here we have ammonia on the bottom, and it can either come through for diffusion or it can also be done for nitrate, and there is also enzymes. So for example, for nitrate, to become ammonia to be incorporated into proteins, this needs to go through nitrate reductase and nitrite reductase before it can be incorporated. And these enzymes also have different trace metal cofactors. So this sort of, we wanted to ask the question, how does nitrogen form and micronutrient limitation affect the growth and toxin production? And so we hypothesized that nitrogen form and micronutrient concentration would interact to affect the biomass and microcystin production in microcystis populations. And we predicted that microcystis grown with urea would actually be able to produce more biomass than cultures grown with nitrate. And that's because urea contains 2 nitrate molecules for every one. So for every urea that is transported through the cell, it can generate 2 ammonia compared to every nitrate where it can only generate 1. And so what we did is we acclimated Microcystis aeruginosa LE3 that was isolated from Lake Erie to low micronutrient conditions. And the micronutrients that we used were just in the BG11. So this was like iron, molybdenum, boron, cobalt, zinc, the general micronutrient media concentrations. And we acclimated them to low micronutrient concentrations. And then we exposed them to 1 phosphorus level and two N to P levels, 4 and 100. And just for ease, we're just going to call this low and high N. And two forms of nitrate, urea, and two forms of nitrogen, urea and nitrate, and then the micronutrient concentrations, low and high. And we grew these for 14 days, and we measured particulate microcystin LR and C to N stoichiometry and a bunch of other things that I'm not going to discuss. And this experiment was part of a course-based research experience that was a cure that was led by Thad Scott and Jackie Duke here at Baylor. And here's all our undergrad co-authors from this paper. And so just jumping right into the results, looking at the biomass and stoichiometry, I'm going to walk you through sort of slowly on this first graph because they're all going to look like this. So on the bottom here, we have on the left side, we have the nitrate, and on the right side, we have the urea. And then we have low and high concentrations. And then further, the first bar that you'll see is the low micronutrient, and the second bar is the high micronutrient conditions. And so this top graph is biomass, and this bottom graph is C to N stoichiometry. And so what you see here for biomass is, and this is just looking at the low N conditions for both nitrate and urea. So biomass, micronutrients or nitrogen form, they weren't affected. However, the C to N stoichiometry was. So in nitrate conditions and low nitrate conditions, adding more micronutrients actually decreased the C to N ratio. And this is actually, we found the opposite with the low urea conditions, where adding more high micronutrients actually ended up increasing the C to N ratio. And then jumping over to the high nitrate and high urea conditions. So micronutrients actually did affect the biomass produced, more so for urea than for nitrate. So in nitrate, micronutrients didn't overly affect them. But yes, for urea, we had the most biomass produced in high micronutrient, high urea conditions. And then looking at the C to N stoichiometry, the high micronutrients didn't, the high or low micronutrients didn't affect the C to N ratio. And that was also regardless of nitrogen form. So then to look at the nitrogen, particulate nitrogen concentration, and also the microcystin. So again, we're just looking at the low, and then the first bar is the low micronutrient and the high micronutrient, and this is nitrate and urea. So particulate nitrogen, there was no difference. And the microcystin concentration, there was no difference. And I'm showing concentration here, but cell quota followed this similar pattern, and we didn't find any difference in cell quota as well. However, looking at the high nitrogen concentrations, again, similar to with the biomass, we actually found the most particulate nitrogen in the high urea, high micronutrient, and micronutrients did affect the particulate nitrogen. So yes, increasing micronutrients, increased particulate nitrogen. Another finding that actually was slightly counter to what I expected was that nitrate microcystin LR concentrations was actually increased compared to the urea. And this was also the case for cell quota concentrations too. And micronutrients didn't affect the concentration. This was just dependent on the nitrogen form. And so sort of to just wrap up this, so high urea conditions did produce more biomass. The energy, potentially could have been less energy to convert urea into ammonia, and that's because there's less uptake transporters, and also the carbon from urea potentially can also be used as a carbon source. In high nitrate conditions, they were more toxic. And then doing a literature search, it seems like the different nitrogen forms can actually result in different amino acid concentrations, and then changing the supply of reactions needed to synthesize the microcystin LR. And so overall, the stoichiometry, the concentration, and the nitrogen form can actually affect microcystin cell quotas. So let's just go back to this hypothesis of cyanotoxin production. So since 2014, another meta-analysis has been done by Brandenburg and others in 2020 that looked at sort of more different data. So they included more studies. And again, they had the saxitoxin or PSP from the previous study, microcystin, and now there's actually been a few studies on cylindrospermopsin, the CYN. And so this again is a log response ratio where we take the treatment and you divide it by the control, and this is all laboratory studies. So what you can first notice here is there's a fair bit of studies done with the saxitoxin, which was mainly dinoflagellates, and microcystin, but there's been very little information known about cylindrospermopsin and how it responds to nutrient changes. And this new meta-analysis found that nitrogen limitation actually didn't result in a decrease of enriched cyanotoxin concentrations or cell quotas, but phosphorus limitation did result in an increase of microcystin and saxitoxin cell quotas. And so when we look at what cylindrospermopsin can be produced by, these can be produced by two filamentous cyanobacteria, Cylindrospermopsis, which is where cylindrospermopsin was first identified, and also Aphanosomena. And both of these species are unique in the fact that they're diazotropic. And as I mentioned, diazotropic means that they're able to fix atmospheric nitrogen. So atmospheric nitrogen is generally inaccessible to most organisms. However, diazotrophs have this enzyme called nitrogenase that converts atmospheric nitrogen into 2 ammonia molecules. And this process is energetically costly, using 16 ATP to generate those 2 ammonia. And additionally, nitrogenase is inhibited by oxygen. So photosynthesis would inhibit the nitrogenase activity. So different cyanobacteria have different different adaptations in which they use so that they can actually fix nitrogen. And so both cylindrospermopsin and Aphanosomena produce these heterocytes. And what they do is, so once the combined nitrogen source, so they'll prefer to use nitrate, nitrite, or urea, ammonia, because it takes less energy. But once those are exhausted, they produce these heterocytes within 24 hours. And these heterocytes, they don't photosynthesize, and they have thicker cell walls to prevent oxygen from being diffused through. And then they are where the site of nitrogen fixation happens, and then they shuttle the newly fixed nitrogen to the other cells. And then if nitrogen is reestablished within the system, nitrogen fixation stops, just because it is that energetically costly. And depending on the cyanobacteria, those heterocytes can either remain on the filament or they can be discarded. And so going back to this, looking at the cylindrospermopsin, we wanted to sort of look at how cylindrospermopsin production varied, particularly with nitrogen limitation. And we would hypothesize, just from the data out there, is that the N-to-P supply doesn't affect the cylindrospermopsin cell quota. And so what we did is we grew Aphanosomena flossacque from France under a range of N-to-P from 1 to 100, and we replicated that 4 times. We used 1 phosphorus treatment, and to generate the N-to-P, that resulted in nitrate added between 0.16 to 16 milligrams per liter. And then we measured the elemental composition and also the particulate and the dissolved cylindrospermopsin with the mass spec. And then to look at the particulate nitrogen here and versus the supply. So this is how we looked at nitrogen fixation. So this is just total, it's not in a rate. So this is using mass balance. And so on the x-axis, we have the supply. So this is the N-to-P1 with 0.16 milligrams per liter, and then the N-to-P100 with 16, and the particulate nitrogen we found in the jar. And so data that falls below this 1-to-1 line means that there's still nitrogen in the media and there should not be N fixation happening. And data that falls above this 1-to-1 line means that the only way nitrogen could occur in the jar is through nitrogen fixation. So then adding on the data here, we see cultures below an N-to-P of 20 actually had nitrogen fixation, and they are labeled in the white dots. And then cultures in which still had nitrogen in the media occurred in N-to-P 30 and above. And then, so to look at the stoichiometry results here by N-to-P. So again, now we have the N-to-P by resource on the X, and then the C to N by mole on the Y. Compared to the microcystin results, you can see that these are really a lot more constrained compared to the microcystis results that we had. And again, the fixation are in the open circles, and the ones that weren't fixing are in the closed circles, the blue circles here. And we found that the NDP 1 to 4 treatments had higher C to N compared to the other ones, and 8 to 20, even though they were fixing, they still were able to maintain similar C to N ratios. So then looking at the cylindrospermopsin results, so this is total cylindrospermopsin. So this is again a concentration by resource of what the N to P was. And we find this segmented linear regression that has a breakpoint exactly where the nitrogen fixation stopped. So below 20, we ended up with decreased total cylindrospermopsin, and above that, it was slightly more increased cylindrospermopsin. However, since we know that nitrogen fixation is a really energetic cost and it causes a growth trade-off, we need to really standardize these results. So looking at the cell quota, we actually find the same pattern. It's not as drastic, but again, the breakpoint was 20, exactly where nitrogen fixation happened. And below that, we have lower cell quotas of cylindrospermopsin, and above 20 with no nitrogen fixation, we have more or less of a stable cell quota happening. And then similar to the microcystin results, we just sort of wanted to look at how the cylindrospermopsin and the C to N correlate to each other. So this is an x-axis in which we have the C to N, and the y-axis we have this total. And again, you end up with this segmented linear regression in which C to N ratios above 6.9, you actually end up with a slight positive slope, but more or less stable cylindrospermopsin concentration. Whereas really low C to N ratios, you end up with these high cylindrospermopsin concentrations. And since this total is contained by particulate and dissolved, just looking at those independently, here is the particulate. So this is found within the cell of cylindrospermopsin by the C to N. There's a lot of variation around this line, but we do have a significant R squared of 0.15, in which there is still a negative relationship. So again, higher C to N produces lower amounts of cylindrospermopsin within the cell compared to lower C to N. And then looking at the dissolved C to N and dissolved, or the dissolved cylindrospermopsin, sorry, versus the C to N, you get this really striking pattern. At above 6.5, 6.6, we actually end up with no dissolved cylindrospermopsin found within the cells. This is definitely not what we expected, and a lot of dissolved cylindrospermopsin found within the cultures of really low C to N. And you can, this pattern is the same if you did percent of dissolved versus instead of doing it by cell quota. And upwards of 50% of the total toxin is dissolved in these really nitrogen rich. And so just sort of finishing this off, nitrogen fixation doesn't completely set off N limitation. There is still signs of N limitation, and that is especially observed in the cyanotoxin production. So cylindrospermopsin production does change in response to the N to P supplies, unlike what the meta-analysis found. And that, at least for RA phantasomina, and nitrogen limited blooms are less likely to be toxic by total concentration and by cell quota. And also the differences between the particulate and the dissolved. So low C to N ratios really increase the likelihood of dissolved cylindrospermopsin that can make it harder to treat water. And then, so I just wanted to wrap up with sort of a, how we can think to begin to generalize all this data into sort of a synthesis of what's happening ecophysiologically. And so here is my conceptual cyanobacteria cell, and looking at sort of excess nitrogen conditions compared to growth demand. This can either be in phosphorus limited conditions, and if that's the case, we would expect if there is any sort of polyphosphate storage to be catabolized from and being used for primary metabolism and for growth and light harvesting like phycobilin proteins. And then if there is excess nitrogen, this excess nitrogen can go into the secondary metabolism. And the secondary metabolism, this could either be storage of cyanophycin, that is arginine and leucine polymers, or cyanotoxins such as microcystin, cylindrospermopsin that we have been working with. And additionally, the other storage molecule that our lab has recently identified by our PhD student Jin Yu, that was just published in LNO, is these light harvesting phycobilins can also act as a nitrogen storage molecule. So potentially there's places where this excess nitrogen can go. And so what I think might be happening here is there's this potential trade-off between growth and storage. And so here on the y-axis, we have a nutrient growth to storage ratio. And on the x-axis, we have a cell quota. So minimum cell quota versus a maximum cell quota. And each one of these storage components actually does have a minimum and maximum cell quota just based upon the literature. So when the cells are growing really fast, they have a high growth to storage ratio, meaning that most everything is being used for growth. And then there's the minimum quota for these storage compounds. And then uptake can exceed growth demands, whether that's just due to differences in uptake or limitation by another element or light. And that will cause the growth to storage ratio to decrease, adding on, sorry about that, my cursor isn't moving, decreasing along the ratio and increasing in the cell quota. And then once other factors truly become limiting, you can get really low nutrient growth to storage ratios, and that's a lot of cyanotoxin production can occur. And so we're really trying to look through this and sort of find if there's truly is data to back this up. And we have a little bit using this phycobilin, and we do have cyanophycin data, but however, we haven't completely analyzed that data set yet. However, looking at the phycobilins, so these are light harvesting molecules that can also be used as nitrogen storage. In both the cylindrospermopsin experiment we did, and also with microcystin that are both enriched cyanotoxins, we find that they're positively correlated with phycobilins. So from our cylindrospermopsin experiment, here's the phycobilins in picograms per cell, and the total cylindrospermopsin, and we see this linear relationship. And then a positive linear relationship. And similarly with phycobilins and microcystin. And this one is standardized to carbon because we didn't have cell quota data there. But basically, we're seeing the same sort of pattern that as you increase phycobilins, we know that those phycobilins can act as storage molecules, and these increases occur in high nitrogen concentrations. You're also seeing this increase in cyanotoxins. And so this idea that this storage cyanotoxin growth trade-off is potentially becoming likely, and I would really like to discuss it with everybody who is listening right now. And so just to summarize, these enriched cyanotoxin concentrations in cell quotas definitely are correlated with nitrogen and phosphorus. And these concentrations, again, are correlated with C to N as we've been finding. And different nitrogen forms we need to sort of pay attention to because they can result in different cyanotoxin concentrations. And this idea that there's this end storage cyanotoxin growth trade-off relationship we're trying to work through as well. And with that, I would like to thank our funding sources from our NIH Center and the Scott Lab with all of your help filtering and culturing all of these organisms, and the Brooks Lab, especially to Reagan, who has ran many of our cyanotoxin samples. And I will take discussions, questions, whatever we have.
All right, Nicole, I'll ask a question.
Thanks, Pooney.
I'll try to find my video. There you go. Hey, awesome stuff. Very cool. Yeah, I kind of missed the when you said the error in each of the phosphorus levels was due to nitrogen levels. This is early on in the talk. You said it's likely, so I was kind of. Can you explain?
I mean, it most likely is. I can't see what else it would be. I mean, because we have. It's the N to P range between 1 and 100. So a lot of those were N limited. And so therefore the biomass just was less. So yeah, I can't see what else it would be.
Okay, so I'm trying to get how that graph is organized.
If I can.
That is, so all of those stacked points are different n levels per.
Yes,
level.
yes.
Okay, got it.
I can
Okay.
try to share my screen again, but I don't know if it's gonna. I'll just bring. It's working on it. There we go. So yeah, so these, you guys can see my cursor, right?
Yep.
Okay, yeah, so like this is like the end of P1 and this is the end of P100. Again, P1 and P100. So yes, so there is some differences based upon nitrogen. Yes.
Thank you.
Okay.
Okay, I'll go, Nicole. Can you go back to your sort of conceptual slide, like looking at the urea and the nitrate coming in?
Yeah. Just trying this one.
Yeah, I mean, I thought that that was just like such a really cool story, and I know that you said like what you thought was going on there, but I was wondering if you could go back through that because I think it's a really interesting trade-off between the the inorganic versus organic. We normally don't think about those. It's normally, we're thinking about that going through there. And do you know what, I'm just trying to think, I guess I got two questions, like energetically, which one is more difficult to change because from like, to incorporate nitrate or urea? Like, I guess that's one question.
So the actual energetics of it, I believe nitrate is more energetically unfavorable, and that's just because of it has to go through nitrate reductase and then nitrate reductase. So it takes more enzymes and more irons and molybdenums needed to make it ammonia. Whereas urea just uses one urease and it can create two ammonia molecules. So it's getting double the bang for each enzyme activity that it turns.
Yeah. That's really cool. And then so you think that but you said your prediction, what you would have thought of happened, didn't. So because like I'm thinking energetics and how that might happen. So
I was thinking because urease it's giving 2 for 1. So for every molecule that it's doing, it's potentially less energy. So then you could potentially produce more microcystin, but we found that nitrate produced more microcystin, either concentration or cell quota. And I think it has to come down to with the literature, and they sort of, even though it goes to ammonia and then it goes through this GOGAT pathway, the glutamine pathway, it seems like there is still different synthesized amino acids. And so when you're growing on urea, you're not producing as many lysine and arginine, which are essential in microcystin, especially in microcystin LR, which that LR stands for, or leucine arginine. Whereas if you go through nitrogen, you are producing nitrate, you are producing these more leucine and arginine type molecules that can be easily incorporated into microcystin.
Thanks. I didn't get that from before, but I knew that you would be able to walk me through a really complex metabolic pathway and then very, simple ways where I could understand. So yeah, thanks. That's
problem.
Really cool. Are there different, like in all those processes, like are the enzymes involved with those? Like, I'm sure they probably have different sort of trace metal cofactors and stuff as well that could be interacting with those.
Yeah. So urease uses nickel. It just has 1 or 2 nickels within its cofactor. Nitrate reductase uses molybdenum and iron. And then nitrite reductase just uses iron. So there definitely is going to be different trace metal factors involved in the production, like using urea versus nitrogen or nitrate. Sorry.
And then further down the line, when you're making your cyanotoxins, I'm guessing, like, yeah.
Yeah. So I was surprised because I thought, like, as soon as you get ammonia, then you can make whatever you want. But there has been, as I said, papers out there that seem like it doesn't just necessarily, it makes whatever it wants. It actually still does have like different amino acid compositions. So then it potentially can affect the amino acids available to make the microcystin molecule, or they're all sort of amino acid based derived.
Awesome. Yeah, I was just thinking, like, whatever stitches those amino acids together to make the microcystin protein, like, are those enzymes, do they potentially have
so I don't know
that are the same or different, you know, as those pathways? You know what I'm saying? If they're trying to
break
down
I totally
nitrate, and this is super iron rich, and those enzymes have a lot of iron, then they might be banging into one another, which is why you didn't see what you thought you might too.
That is actually a really good point. I actually don't know the micronutrient concentrations or like the demand of micronutrients for the microcystin genes. So there's a bunch of microcystin genes that go into making a microcystin molecule. They are all the same, whether depending on like the LR versus there's a bunch of different congeners. They're all the same. But yeah, I don't actually know, but that is a good point that it could.
I wasn't trying to trip you up. I was just trying to think of something else.
No, that's a good thought. So no, that's perfect.
Thanks.
Hi, Nicole. Great talk. This is Chris. I have a question related to the C2P stoichiometry that you saw in some of your treatments.
Yeah.
How did the flexibility of that compare to what's previously been shown in the literature? You know, I'm kind of thinking about some of Jim Kotner's work, but that was mostly on heterotrophic bacteria, if I'm correct. And so I was kind of wondering how that translates to autotrophs.
So from a phytoplankton point of view, we would expect this range of plasticity, just given the different amounts of phosphorus. But from a bacteria autotrophic point of view, I think this is really more plastic than what people expected. I know I have grown Seneca coccus, and I couldn't get that above like 500, the C2P, no matter how low of phosphorus I went. So yeah, I think the microcystis has a lot of plasticity there.
Okay, thanks. Yeah, it seemed like a much bigger range than I thought it might be. So maybe what you said is exactly right, and I just wasn't thinking about it in those terms. But yeah, thanks a lot.
Yeah. From a phytoplankton point of view, we would expect this, but from a bacteria autotrophic point of view, I think the literature, they're a lot more constrained. If other people have any ideas, though, jump on in.
Hi, Nicole. My name is Paul Juckelman with the U.S. Geological Survey, and one of the areas of research that I'm focused on is actually trying to help with nutrient reduction plans, agricultural work.
Yep.
So not being a biologist, I'll admit I understood maybe 1% of what you presented, but I want to make sure that I understand the take-home message for someone like me properly. Societally, we want to reduce both the mass of algal blooms, but also the toxicity.
Yes.
And so it seems to me that if I'm understanding this correctly, by focusing first on phosphorus reduction, we could potentially decrease the biomass while simultaneously increasing the toxicity.
Yes, that is exactly what potentially could happen. So then I guess your total toxin concentration will be less, like within a lake if you add up the toxins, but on a per cell basis, they could actually be more toxic. So we need to be mitigating both phosphorus and nitrogen to not only reduce the biomass, but also reduce the toxicity potential of these HABs.
Great, thank you.
Yeah.
So maybe we should just leave it there and thank Nicole for giving a great talk. This is really enjoyable. It made me really proud as her former PhD advisor to see all this great stoichiometry. And so thanks again, Nicole. Thanks for everybody for coming. And I'm sure if you have any questions, you can just email her.
Yeah. Email me. It's Nicole_Wagner at Baylor. wagner.edu, or if you don't remember that, you can email Ratios Matter. I can get it.
All right. Thanks, everybody.
Well done, Nicole.
Thanks, Clay. Caleb's probably not paying attention if you want to say something.
No, yeah.
Oh, there.
So that was really good. Like,
Thanks.
um,
Oh,